Thank you. Thanks, Danny, for inviting me here. So this has this complicated title about membrane fluidity domain formation. What I really want to explain is kind of the other lipid types, lipid domain types that most of you guys probably don't hear frequently. So I'm not going to talk about lipid rafts. I'm going to talk about all the rest of the domains. And before I forget, all the experimental stuff that I'll be showing is with Bacillus subtilis. It's the gram-positive model organism. Doesn't have an outer membrane, which makes life so much easier when you're working with membrane stuff. And it's also, as Danny said, very beautiful. So before I want to start, I want to introduce like 3 basic concepts related to membrane lipid domain organization. Probably you know most of these, but still good to wrap it up. So first concept is membrane fluidity, because you'll be hearing a lot me talking about the membrane fluidity. So what that really means that there are 4 different kind of parameters in a membrane. There's the lipid head group packing, the fatty acid disorder, and the ability of lipids to laterally and rotationally diffuse. So those 4 parameters of lipids are very tightly interconnected. So usually when one is changed, the other ones are changed too. And these together, these 4 tightly interconnected membrane parameters, we call membrane fluidity. So it's not necessarily a physical parameter by itself, but it's and to describe this phenomenon. So basically what we mean with this is that membrane can come with a different configuration. They can be less fluid or more ordered, more tightly packed, where the lipids are diffusing less, or they can come in a more disordered, more loosely packed configuration where the diffusion is faster. This is what we call more fluid, this is less fluid, and really everything in between, what configuration the membrane takes, depends on a lot of factors like temperature, fatty acid length, saturation, branching, the lipid head groups, presence of membrane proteins, and intercalating molecules like cholesterol, hopanoids, and carotenoids. So this by itself is important when we understand lipid domain formation, but the other phenomenon is lipid phase behavior. So basically, in addition to showing different fluidities, membranes can also adopt different type of configurations where the transition from one to another one is quite abrupt. So membranes can come in so-called chill phase, where the lipids are really tightly packed, have low fluidity, and these are kind of promoted by saturated fatty at this low temperature. But these are not normally present in biological membranes, at least are not thought. Then it's the liquid ordered phase, which is also tightly packed and more fluidity. But these are induced by compounds like cholesterol and carotenoids and hopanoids. And these are part of biological membranes as lipid rafts. But the last, and for my stuff, the most important lipid phase is the liquid disordered phase. So these lipids are relatively loosely packed. But what is important here is that it's not one configuration. Within this one phase, the membrane fluidity can have vastly different values. So it can be relatively low fluidity or very high fluidity, again, depending on stuff like temperature. So these are induced by unsaturated branched-chain fatty acids and high temperature, and this is kind of regarded to be, at least in bacteria, kind of the regular state of biological membranes. So what is important here when it comes to the lipid domain formation is that these are quite abrupt changes from one configuration to another one without big transition in between. But there are other types of domains too. So when we talk about lipid rafts, for instance, this is kind of thought to be lipid phase separation domain formation. So the cholesterol and other parameters cause the lipid to adopt this different configuration, and that can have some area of the membrane and not all. But then there are also fluidity-driven domain formations where just some areas of the membrane show different level of membrane fluidity. And here we're talking about, I will show examples with the lipid domains associated with the bacterial actin homologue R4B. And at last, we can have lipid head group-driven domain formation. So this is the first type of bacterial lipid domain that was characterized. So it's called the Cardiolipin domain, which was shown to form clusters. So this is not really necessarily related to fluidity, but different local head group composition. But of course, it's worth mentioning that this is all very simplistic, so it's not so black and white. You can have this kind of domains that also have a different fatty head group composition, and lipid rafts don't necessarily have the same head groups than the other ones. So these are more like conceptually what kind of different driving forces can be there to promote domain formation. So let's talk first about the cardiolipin. So this is the example of the lipid head group driven domain formation. So in bacteria, among phosphatidylglycerol and phosphatidylethanolamine, cardiolipin is one of the most common head group phospholipid species. So in E. coli and Bacillus about 5 to 10%, but can reach up to 50% in Staph aureus and strep pneumonia. And these lipids are postulated to form specific lipid domains at cell poles, which are implicated in various cellular processes like cell division, DNA replication, chemotaxis transport, antibiotic mode of action. A lot of things have been at least postulated to be linked to these polar cardiolipin domains. So what really pushed further this, where really this concept started was actually from two observations. First, there was the ability to stain them. So there is a dye called nonylacridine acridine orange, or NaO, which is a positively charged membrane dye, and that shows an emission shift from green to red fluorescence, which is supposed to be specific for when it's interacting with cardiolipin. So here you can see examples from all the papers. Well, let's actually review these ones, but you can see polar localization pattern in E. coli and here at the cell division site, and these were interpreted as membrane domains characterized by local cardiolipin enrichment. Why this was so well accepted as a concept was because of actually very neat experiments. So in E. coli, the normal cell division happens between the nucleates, and there are two machineries that take care that it actually happens there. There's LIM-A that prevents cell division to happen over the nucleate, and the MIN system that prevents it to happen in poles. You get rid of the MIN system, and E. coli can undergo cell division here, and it forms so-called mini-cells, which are basically just a cell pole being divided out of the cell. So if what they did in this paper was that they analyzed the fat lipid composition in wild type and compared it against mini cells. And what they showed here is that there is a distinct enrichment of cardiolipin in that polar area, which was fully consistent with the staining pattern. And this is basically what was seen as the independent proof for the presence of polar cardiolipin, and everything was happy. And after that, in a lot of other bacteria, these cardiolipin domains have been described. But what is actually important here to keep in mind is in all of these, it's just based on NaO as a staining. These polar mini-cell experiments never been repeated in any other organisms. So 2014 came a little bit of a question here, because this was from a group of Doug Weibel, where they showed that actually, while we see in wild type these polar domains, we also see them in a strain, also they see in a strain that doesn't have cardiolipin at all. And it only takes deletion of PGSA, which is responsible for synthesis of PG, from which cardiolipin is also made, that these polar domains disappear. It's just quantification of it. So a quote from their paper: although several other groups have successfully used micromolar concentration of NaO labeling studies, we found that the polar enhancement of fluorophore is unreliable at concentration above 2 micromolar, below 2 micromolar. So this is important, because there's a controversy going on that field, where we are now actually contributing to. The one group claims that if you use these higher concentrations, it's not specific anymore, so therefore they see staining in absence of cardiolipin, whereas the others don't see any domains at lower concentrations and only see them with these higher concentrations. This is actually what we also see. This is still also all in E. coli, so we started to look at this Bacillus. And this is when we stain Bacillus with this membrane dye. So important, we are always looking at two different wavelengths. We are looking at the green and the red wavelength. The red wavelength should be the cardiolipin-specific signal. And green should be just for negative phospholipid in general. But in both cases, in LB and in DSM, which was used in an original medium where Bacillus cardiolipin domains were postulated to be, we don't see any domains. So that was the first surprising for us, that actually what's going on in there. So we went more carefully repeating the original experiments from the paper. And one of the key differences, we found that actually we do our experiments, when we grow the cells in 37 degrees, we also stain the cells in 37 degrees, whereas in the original paper, the staining was described to have been done in room temperature. So we repeated the experiments, but this time we incubated the same 20 minutes like in the paper in 20 degrees instead of the growing temperature. In LB, we still don't see anything, but now we see in this DSM medium these domains that look very much like what was published earlier. So we have two possibilities actually, and we were in discussion with the original authors about this too. So without cold shock, we see stronger septal staining at septum. So there is a possibility that this is actually the cardiolipin signal, or we see only when the cells undergo this cold shock, we see these domains, and this could be the cardiolipin domains. So, but the problem with the septal thing is that actually this is where the cells divide. So we have two membranes present there. So we did some calculations and measurement of the fluorescence intensity, comparing the signal measured of the septum and across the septum, where we know that, okay, this is just one membrane, with the expectation that twice the signal at septum would be just coming from these two membranes present. And we're actually not even getting it twice, we're getting 1.6 times. So this septal signal, it's just purely a matter of membrane geometry and nothing to do with any specific lipid localization. So how about these domains that we see upon cold shock? So luckily cardiolipin is not essential in bacillus, so we can delete the synthesis for cardiolipin, and this is just TLCs showing that, yeah, cardiolipin is completely gone, and we can repeat this experiment, and the domains are still there. So these domains that are here are absolutely not characterized by cardiolipin. They can only also emerge in the presence in absence of cardiolipin. So therefore, these cold shock triggered domains are also not cardiolipin independent. So the last one, and this is specific about the question of the concentration. The one E. coli group claims that this is kind of the range that should be used, otherwise it's not specific. The other group claims that this is where everything needs to be done, otherwise there are no domains. So we did the same in bacillus, we did the concentration range, and indeed when we use higher concentration, we get polar lipid domains that look very similar to what's seen in E. coli. But unfortunately, this is where the dye starts to become growth inhibitory. So it's quite worrisome that the domain formation only happens under conditions where the dye is actually lethal. But either way, even on those higher concentration, it actually has nothing to do with cardiolipin. So also in absence of cardiolipin, they're still formed. So comparable to E. coli, higher concentration of now lipid domains can be observed, but these are not specific for cardiolipin and coincide with toxicity. So what we think is that under normal steady growth conditions, Bacillus subtilis membranes actually do not contain lipid domains that can be visualized with NAO. Upon cold shock and when used in toxic concentration, NAO does stain lipid domains of unknown identity, but these are cardiolipin independent. And in our hands, NAO really doesn't exhibit any detectable specificity towards cardiolipin in Bacillus membranes. So we think that there really is no reliable evidence for domain formation of cardiolipin in Bacillus. So this is all published, so if you're interested, you can look at more details about this in that paper. So this is now an example, a little bit of a negative example with this lipid head group situation. But let's go for the fluidity-driven domain formation. Rotate and move in tracks that are perpendicular to the x-axis. What this does is, the current model is that this drives the cell wall synthesis to new peptidoglycan strands to be incorporated perpendicular to the cell axis, as visualized here by fluorescent vancomycin, which stains areas where the new cell wall synthesis takes place. And as a consequence, this machinery allows the cells to grow in length and determine the morphology, so the rod-shaped morphology. So therefore, in the presence of MREB homologs, MRE bacillus is nice rod, whereas in the absence, it becomes this kind of very twisted coccoid organism. So what we showed ages ago is that the localization of MREB is actually dependent on proton-motive force. So when we collapse proton-motive force, or even specifically membrane potential, the protein gets delocalized and it runs from this normal localization pattern into foci in the membrane. And what we realized quite early, that if we stain the cells with membrane dye Nile red, it stains these same foci. So what we thought originally is that if we discover what these are, that's going to explain the mechanism why this gets delocalized. But it actually turned out to be the other way around. So if you look at the bacillus have 3 MREB homologs. If we delete every one of them individually, the foci still emerge. If we treat the cells with CCCP, which collapses the BMF, but in the absence of MREB homologs, nothing happens. So it looks to be the other way around, that it actually needs the MREB cytoskeleton. So what are these membrane irregularities? And this is, first, conceptually, there are 2 possibilities when you have this kind of thing. This is a possibility that's actually quite commonly overlooked. So if you have a focus in a membrane with a membrane dye, that could just as well be actually a local invagination from the membrane, because given the low resolution, this kind of structure would just see like a splotch in the membrane. So we need to distinguish whether it's an invagination of a membrane or actually a lipid domain. So luckily, if we compared this localization pattern with membrane proteins here using ATP synthase, this protein isn't delocalized, and it reports nicely smooth membrane in the areas where we see these membrane invaginations, kind of telling that it's just not continuous membrane that is continuous, but it's something specific in these membrane areas which isn't just physical deformation. So what could these be? Again, we tested cardiolipin, strains that don't produce cardiolipin. Phosphatidylethanol is also not essential in bacillus. We could delete the synthase, they are still formed. Phosphatidylglycerol is essential, so we could deplete it. We still had no quantitative difference in the foci emerging. Lysyl PG glucolipids, we also got rid of them. They are still emerging. It doesn't really look like caring much about the head groups. So, but we still see this with Nile red. So Nile red is a neutral hydrophobic dye, but it's actually sensitive to fluidity. So what I'm showing here is bacillus cells. So Nile red is non-fluorescent in solution, but when it incorporates the membrane, then the fluorescence picks up, and this is something we can measure just in a fluorimeter. And what you are looking at here is measurement in 30 degrees of cells that are grown in 30 degrees, 48, or in room temperature. So basically, these are dropped to lower temperature, which would cause the membrane to become less fluid and more rigid, and these are increased in temperature, which will make the membrane to be more fluid than cells that have adapted to it. And we can see this nice gradual change in the staining intensity, implying the Nile red is not necessarily preferring, but it's definitely, it could be either showing higher fluorescence in more fluid membranes, or it could just accumulate there better. So anyway, it's actually sensitive to membrane fluidity in vivo. So could it be that these are these kind of fluidity-driven domains where we just have areas that are characterized by looser packing? So, and what we use for this, to address this question is Laudan. So Laudan is a membrane fluidity-sensitive dye, which incorporates in a membrane roughly like this. And what is really sensitive to is water. So its emission spectrum is sensitive to water and how water penetrates in a membrane. So in a tightly packed membrane, water is less prone to penetrate, whereas more loosely packed, there is more water in the membrane. And this changes the fluorescence behavior of the dye. So in a very tightly packed, you get the peak at 445, somewhere in there it is. But we get this another peak appearing, which correlates to the degree of this is the highly packed one and so on. So what you get is a shift in a spectrum that depends on how much water is in there. And by measuring the fluorescence in these two wavelengths, you can calculate something like a Laudan GP, which is a ratiometric measurement, which just simply gives you in a scale of -1 to +1 a relative level of membrane fluidity. But what is interesting in this is that this could also be combined with microscopy. So we stain the cells, we take pictures in those two different wavelengths, and then pixel by pixel we calculate this Laudan GP value. And here you see that when we treat the cells with CCP, we are getting values that are correlated with higher membrane fluidity values. So lower membrane Laudan GP values. And these are actually co-localizing with Nile red. So the same foci that we're seeing with Nile red, we are seeing with Laudan as being areas with increased membrane fluidity. So Nile red stains domains with elevated membrane fluidity, and this is what Leonard coined regions of increased membrane fluidity, or RIFs. It's probably a term that you will encounter many times when he's talked to. So, but the problem here is this is something we see in under conditions where we throw CCP on the cells. There's a question of, are these always present, or are they only caused by this delocalized MRAB? And actually there we turned back to in vitro or more eukaryotic origin of dyes, and this is actually a lipid mimicking dye, which is commonly used in vesicles to study lipid raft formation. So this kind of similar kind of image where Ilya was just showing. These are the liquid ordered phase and liquid ordered phase, and this dye shows preference to it, which is really based on the hydrophobic mismatch of this hydrophobic part. It just matches better to the thinner, more fluid membrane than the thicker lipid raft membrane. So that's a dye that has preference to more fluid membrane areas, and indeed it turned out that when we stain bacillus with this one, we can see a localization pattern that looks very similar to what we see with MRAB. And importantly, if we take a longer variant of it with the mismatch, where the mismatch isn't there, we are losing this preferential localization. And it does co-localize with MRAB. Indeed, we do think that MRAB in its native active configuration, it's worth mentioning this dye is no growth inhibitor or anything, is associated with lipid domains that we can visualize with this dye. So, to summarize, the bacterial cell wall synthesis machinery assembled around the MRAB actin cytoskeleton is associated with lipid domains characterized by increased fluidity. And these domains can be preferentially stained with fluorescent dye DISC12 when it's in a native dynamic state, or when clustered, like as caused upon clustering of MRAB, the local fluidity changes can be measured following the backing-dependent emission of spectrum of Laudan. But what is really important here is that this relaxed lipid backing of this type of lipid domains makes it generally easy for any membrane dye to incorporate. Anything that needs to intercalate into this kind of domains, like peripheral membrane proteins or dyes, really likes these kind of domains. So therefore, these can be stained with Nile red, FM dyes, Mito tracker green. Even now is showing very nice preferential localization in those. So I think a lot of the stuff with the E. coli now actually can become from this type of situation. They're actually domains with increased fluidity, have nothing to do with cardiolipin, and now is showing the same behavior as most membrane dyes to integrate in those. So, at last, I want to introduce something that we recently been working on, and this is antimicrobial induced lipid phase separation. So now we're talking about the conceptually the same type of lipid domain formation as in the case of raft. So we get areas of membrane that somehow adopt a different configuration. So I'm keeping this relatively short, although this is a complex story, but this is a small cyclic cationic antimicrobial peptide that literally it's 3 arginines and WFW. This is the whole thing. But what is really interesting in this is that it has a very low MIC against gram-positive and gram-negative organisms. Usually membrane targeting antimicrobials are quite toxic in eukaryotes. That's why there are only few examples of where they're clinically used, like daptomycin and polymyxin B. And what is really promising in this one is it's not hemolytic against human erythrocytes, and it's not toxic against HeLa cells. So it shows quite good initial characteristics when it comes to actually being a potential antibiotic. And what is really cool is that it has very low resistance development, which is actually shared by membrane targeting antimicrobials in general. So we can't detect any resistance development against this one. We know it targets membrane, but when we started this study, we didn't really know what it is. And we're not going to go into detail, but what makes it special is that unlike any other antimicrobial peptide, it neither, so far studied, it neither permeabilizes nor depolarizes the cytoplasmic membrane, yet it does something. So what this something turned out to be is actually quite cool. So when we are now using Laudan as a spectroscopic way to just monitor the membrane fluidity of living cells in their own growth medium. But what we are doing here is not more microscopy, we are just measuring the fluorescent layer and how it changes. So this is a trace that we get over time of 30 minutes where we're monitoring this for untreated cells where the fluidity stays relatively stable. And here we are adding benzyl alcohol, which is a chemical membrane fluidizer, just as a positive control. So important here, low Laudan GP values represent high membrane fluidity and vice versa. So we get this drop in Laudan GP indicating the fluidizing effect. When we add this peptide, very rapidly we get a really significant rigidification of the membrane. And this is actually corresponds to roughly 20 degrees of cold shock to the cells, just by adding it. So it's really going from the normal configuration into one that is much more tightly packed and less fluid. So the question here, of course, was that is this phenomenon what we see, this very quick rigidification of the membrane, some kind of very homogeneous effect on the membrane, or is it something that is actually on a sub level, some domains are formed that show different fluidity and so on. So we turn back to Nile red again, so untreated, this is how it looks, shows no domains. If we treat the cells with five minutes with this peptide, where we get the full plateau in the rigidification. So if you look at here, after 5 minutes, we are somewhere here. So we have the full rigidification. The staining does go down a bit, so this is adjusted for it, but we can't see any domains being formed, which kind of inclined that yes, this rigidification is really quite uniform effect on the membrane. What happens though is with the delay is something really quite astonishing. So we get something that looks very much like what you would expect from this kind of membrane phase separation in vivo to be. So we get membrane areas that show clearly different staining intensity than other areas, not just small foci or domains and so on, really large areas of the cells. So we do think that we get a delayed but massive lipid domain formation that is very close to looking like a phase separation behavior. So if we so what we did here was looking at the fluorescent peptide, or the variant of the peptide with the fluorescent label, and this is showing exactly the same phenomenon. So after 20 minutes, that peptide is actually enriched in certain areas in the membrane, whereas initially it's smooth, and these correlate with the areas that we see with Nile red. So the Nile red stains domains are really domains that are also enriched with this antimicrobial peptide. Again, we looked at, is it cardiolipin? Because every referee would always come with that question first. So no, it's not cardiolipin. In the absence of cardiolipin, it is still happening. And another common question that comes, which is really originating from biophysical works with cationic antimicrobial peptide, is demixing of the membrane to anionic and swittionic lipids. Because this is something that is commonly seen with cationic peptides when they are mixed with liposomes composed of swittionic like PE and anionic like PG membranes, that they somehow demix. So is it that behavior? And it turned out to be not. Phosphatidylethanolamin is the only swittionic phospholipid in Bacillus. We can get rid of it. We have only anionic and neutrals left. We are still getting this same phenomenon, so it's not that type of behavior either. But when we repeat our measurements with Laudan using the microscopy, you can clearly see that these areas are characterized by low membrane fluidity. So in this case, low rather than high, as we saw with memory B delocalization. So it really looks like we get a lipid phase separation into membrane areas that are high and low in fluidity. So, but this is not only affecting lipids, this have a huge consequence on the membrane protein. So if we looked at this as a representative though we showed in the paper, we looked at a lot of other proteins too, is ATP synthase, one APC transporter, and WALP23. This is really a model transmembrane domain, which is basically a repeat of alanine and leucines, and one single transmembrane domain. Fused to GFP, express, and we're using these because they show no intrinsic localization pattern. But if we treat the cells under conditions with the peptide and we get this phase separation, we get that these areas that stain with Nile red are really heavily depleted by all integral membrane proteins. It's like someone has bitten a part of the cell. So it looks like integral membrane proteins are really quite efficiently excluded from these peptide-induced domains. But what was really strikingly different is we look at the peripheral membrane proteins. So these are just 3 proteins with an amphipathic helix. Actually, this is this is POVM, this is porrelation protein that has no function here, but this is just one amphipathic helix, membrane binding amphipathic helix, so peripheral membrane protein. And here we are looking just the MIND and ZEPF amphipathic helixes, the 2 proteins involved in cell division, just the amphipathic helix fused to GFP, because they would normally show a localization pattern. So they are everywhere localized, and in this case, when we follow them, they are actually enriched in these domains that are induced by the peptide. So what looks like we get with this peptide really a spatial separation of proteins in the membrane, where the integrals are here and the peripherals are here, which is certainly, you can imagine how this is messing up everything in the cells. That could well explain why it's so antibacterial. So just to summarize, so this is the first membrane targeting antimicrobial peptide that does not act by de-energizing the bacterial cells. Instead, it uses a combination of 3 mechanisms. So we get this rapid rigidification of the cytoplasmic membrane, which is very likely to directly impair a lot of membrane protein functions just by changing the fluidity that much. With a delay, we get a phase separation of membrane into fluid and less fluid domains, and this causes segregation of membrane proteins into spatially separate areas. And something I didn't have time to show is that when this phase separation happens, we get a drop of key cell wall synthetic proteins like MUR-G and MREB completely off the membrane. That triggers a runaway degradation of the cell wall through cell wall autolytic enzymes, and that's actually the mechanism that causes the cells to lyse and also die. So this delocalization of protein is actually triggering an autolytic cascade that kills the cells. So this is also published, so there are a lot of more details about it if you're interested. So as a summary for this talk, so we've given an example for the lipid phase separation driven domain formation. This is not raft. So here we have it triggered by an antimicrobial peptide. Here we're talking about the fluidity-dependent domain formation triggered by MREB cytoskeleton, but it also can be triggered by antibiotics, and I think Leonard is going to be talking about these further. And then I've shown some evidence about the lipid head group driven domain formation, and that is probably not really as conserved as and cardiolipin specific as was postulated. So I'd like to thank the collaborators. So Leonard is involved in many places. Now he's Amsterdam. He used to be in Newcastle. So the MREB work was done when I was still a postdoc in his group in Newcastle. The peptide work was done with collaboration with the group of Makita Tate from Leipzig Institute for Molecular Pharmacology in Berlin and from Julia Bandau in Ruhr University Bochum. Michaela then went to landed in Amsterdam, and there's a nice exchange for people going around here. And BBSRC and Newcastle University provided from my side the funding for these. And at last, I have a postdoc position available to study those MREB-dependent fluid lipid domains. So if someone is interested on coming to Always Sunny Newcastle, where we have a really, really nice center for bacterial cell biology, where we are the whole building full of microbiology. It's really a cool place to do microbiology. 3 years postdoc, even with Brexit, it's available, and it's starting with early next year. So, thank you.